Before delving into vertebrate diversity itself, it's important for us to understand a little bit about where vertebrates came from, who their closest relatives are, and how they generally fit into the animal kingdom as a whole. So why is this so important to understand before we go into vertebrate diversity? Well, it has to do with the way evolution works. So sometimes evolution produces something that is really completely, entirely new, but most of the time it works by making relatively small modifications to what's already there. So if we want to know how we build up to vertebrates, we need to know what the raw material was, what were the building blocks. And the way I like to think of this is with an analogy to these installments that started cropping up in New York City in 2012. So these were created by a New York City architect when he realized that there were all these phone booths all over the place in New York City that were being almost completely unused now in the age of cell phones. Nobody ever uses a payphone. And so what he did was he converted these phone booths, keeping all of the original structure there, into these community lending libraries. He just installed shelves around the structures that were already there and fitted them with books, and now it has a new function. So how is this analogous to the way evolution works? Well, basically it's just building upon the structure that's already there to allow the structure to take on a new function. This is exactly what evolution does, and we'll see this analogy coming into play as we look at the structures in the relatives of vertebrates and see how those were modified to become the structures in vertebrates. As you may remember from previous biology classes, vertebrates are members of the animal kingdom, and they are the largest part of the phylum Chordata. But that information doesn't really tell you very much about who the vertebrates are, who are their closest relatives, and where do they really fit in animal life. So reaching back to what we learned in Gen Bio 2, we're now looking at a cladogram of a clade known as coelomates. So coelomates are joined together by the synapomorphy, the shared derived character of possessing a coelom, which is that fluid-filled body cavity potential space that separates the inner tube of the body, which would be the digestive tube, from the outer tube, which is the body wall, and allows those two regions of the body to develop and evolve separately. And so among coelomates, the first branching that we see within coelomates is between this group, the protostomes. So protostomes include insects, mollusks, annelids, the segmented worms, most of the groups that you would typically think of as invertebrates. And remember, invertebrates is in quotes because that is a paraphyletic group. And then on the other side, this group called deuterostomes. So the first split within coelomates is between protostomes and deuterostomes. And deuterostomes are characterized by several synapomorphies. And we'll go over in more detail in the next slide what these are, but radial cleavage, the blastopore becomes the anus, indeterminate development of cells at early developmental stages, and the presence of pharyngeal slits. So all chordates belong to this group called deuterostomes. Now let's take a look at what all of these synapomorphies of deuterostomes actually mean. So the first one has to do with cleavage. Cleavage refers to the very first cell divisions after fertilization in a zygote. So here we see two embryos at the 8-cell stage, one from a protostome and one from a deuterostome. So one difference between the two is that the protostomes have spiral cleavage, which means that these two layers of cells in the 8-cell stage divide in such a way that they are offset from each other. They're not lined up one right on top of the other, so that if you look at it from the top, it kind of looks like a spiral coming down around the embryo. Whereas the deuterostome embryo at the 8-cell stage, this upper layer of cells lines up right on top of the lower layer of cells. And so if you looked at it from the top, it would just look like 4 cells. The other thing that we see here in this figure is that the cells in the protostomes are determinate at the 8-cell stage. What that means is that the parts of the body that are going to develop from each of these cells is already determined. It's already fixed, so that if you remove one of these cells at the 8-cell stage, the animal is basically going to be missing like 1/8 of its body. It's not going to develop, and that won't be a viable embryo. Whereas for deuterostomes, they have indeterminate development at this stage. The cells are not yet differentiated into what they're eventually going to be, so that if you remove one of these cells here at the 8-cell stage, basically all the other surrounding cells can fill that in, and they have all the information to go ahead and rebuild that cell that's missing, and the embryo will develop normally. Another difference between the two is in the development of the structure called the coelom. The coelom, you might remember from Gen Bio 2, is this fluid-filled sac in the body cavity, a potential space that lines all of the internal organs, and basically it separates the inner tube or the digestive tube from the outer tube, which is the body wall, and allows those two tubes to develop and differentiate and evolve separately. So this is a very important structure. In protostomes, the coelom develops from solid massive cells that hollows out, and we call it schizocele, so it splits. Schizo refers to splitting. It splits to form that cavity. Whereas in deuterostomes, the coelom forms as this out-pocketing, these folds from the archenteron, which is the primitive gut tube. So these are folds from the gut tube that eventually grow out and wrap around the gut tube on either side. And finally, the fate of the blastopore. So if you think back to gastrulation, again, Gen Bio 2, this process of cells from the outside of the embryo migrating inside the embryo and becoming those interior endoderm cells. So remember, we have endoderm, mesoderm, ectoderm. And the little hole that's left after these cells migrate in is called the blastopore. And so at this point where there's just the blastopore, the gut tube is not a complete pathway. Another opening needs to open up at the other end. So in protostomes, that blastopore becomes the mouth. And that's literally what protostome means. Proto means first. Stoma refers to the mouth, so first mouth. And then the new opening that occurs at the other end of the gut tube becomes the anus. In deuterostomes, that blastopore, the first opening, becomes the anus, and the second opening becomes the mouth. So, and that's literally what deuterostome means. Deutero refers to second, stoma, again, mouth, second mouth. So those are the major differences between the protostomes and the deuterostomes. So now let's move up to the next node. The split divides the echinoderms and hemichordates from this other larger group on the tree, the chordata. So these are the chordates, including the vertebrates here. So who are these echinoderms and hemichordates? The echinoderms should be familiar to most of you. They include groups such as sea stars, sea urchins, sea lilies, sea pens, sea cucumbers, sand dollars, things that you can often see on the beach. And these are, of course, radially symmetrical. You wouldn't really think of them as being near relatives to the vertebrates, but it's thought that echinoderms actually evolved from bilaterally symmetrical organisms, and they became secondarily radially symmetrical. The hemichordates include a lot of worm-like species, including the acorn worms pictured here, and the terebranchs, which are a sort of tube worm with these filter-feeding appendages that it uses to collect food particles from the surrounding water. It had been thought previously that hemichordates were actually the sister taxon to the chordates because of the fact that they have these pharyngeal pores, these pharyngeal openings in the front part of their gut tube that perforates all the way through to the outside, and so that's used by some species in filter feeding. They take the water in through the mouth, and then the water comes out through these pharyngeal pores, which is, of course, also a feature in all of the chordates at some point in their life cycle. but research has shown that hemichordates are actually the closest relatives of the echinoderms. The echinoderms probably originally had these pharyngeal pores, but in the course of their transformation from a bilaterally symmetrical to a radially symmetrical group, that they subsequently lost those pharyngeal pores. So that brings us to the chordates. So chordates first appear in the fossil record more than 500 million years ago during the Cambrian period, and they were these very small, elongate, bilaterally symmetrical organisms that we find in the fossil record, some of them quite freaky looking. And something really interesting happened in the first chordates. So if you look at this image down here at the bottom, this is a schematic of a hemichordate. Find that these pharyngeal pores are located dorsally, or on the back side of the animal. And it had a nerve cord that was located ventrally, as it is in most protostomes. Somewhere before the first chordates, there was an inversion of that pattern. The animal was sort of flipped on its back, so that the earliest chordates have those pharyngeal slits ventrally and a dorsal hollow nerve cord. So the positioning of the nerve cord dorsally was hugely important to integration of complicated sensory information and coordination of movement in space. And what seems to have been responsible for that patterning inversion is a relatively simple change in the expression of morphogenic proteins that control ventral versus dorsal development. So it's long been known in vertebrates and other chordates that the morphogenic protein that causes dorsal development is called chordin, named after the notochord, and that the one responsible for ventral development is called bone morphogenic protein, or BMP. And you may remember the story from GenBio 2. BMP is expressed pretty much everywhere. Chordin is an antagonist to BMP that binds it up and causes it not to be expressed. And when BMP is not expressed, then dorsal structures develop, such as the notochord and the nerve cord, etc. It's been found that in hemichordates, the opposite is true. BMP is known as a dorsalizing factor. It directs dorsal development, and chordin is still its antagonist. So where you don't find chordin, you get dorsal development. Where you do find chordin, it blocks the dorsal development directed by BMP and causes ventral development. So a very simple change in the role of BMP and the expression region of chordin may have caused this patterning inversion so that you get structures that were previously dorsal, these pharyngeal slits, now ventral, and the nerve cord, which was previously ventral, now appearing dorsally. So now we come to the first group to branch off within the chordates, the cephalochordates, also known as amphioxus or landslits. And I'm going to use these animals to demonstrate the rest of the synapomorphies of the chordates. So as you can see, the landslit looks like an elongate, really very fish-like organism. They live in the bottom sediments in little burrows. They kind of stick their heads up and filter the water for food. So the way they do that is that they have this gland, this structure called an endostyle, in the floor of the pharynx, which is the beginning part of the digestive system. And the job of the endostyle is to produce a steady stream of mucus that moves constantly from the pharynx down into the gut tube. And so the sticky mucus traps particles of food from the surrounding water and carries it to the gut tube to be digested. So in order to have that steady supply of new water with new food particles into it, there needs to be flow of water through the pharynx, and that is achieved through these pharyngeal slits. So if we look down at this internal view, we see that water comes in through the mouth. It goes out through those pharyngeal slits. So when the water comes in, the food particles are trapped by that sticky mucus the excess water comes back out so that new water with new food material is able to come in. So we call this mode of feeding suspension feeding. Food particles that are suspended in the water are trapped and pulled out of the water and digested. So the endostyle is one of the synapomorphies of the chordates. All chordates have either the endostyle or a structure derived from the endostyle called the thyroid gland. The next structure I'd like to point out is the notochord. So the notochord is a stiff rod that basically keeps this elongate shape in the animal. It keeps the animal from compressing front to back. And the reason for this is that you'll also see that this animal along the back has these blocks of musculature on either side. It looks a lot like fish muscles. They're these little V-shaped muscles, and they're on either side of the body. So the combination of this notochord to stiffen the body and these lateral dorsal muscles allow the animal to move through lateral bending. So if you think about it, if you contract muscles along the body just on one side, and you don't have a rod that stiffens the length of the body's just going to shorten as a whole, right? If there's nothing to resist that muscle shortening. So the fact that you have the stiff rod in conjunction with these lateral muscles allows lateral bending rather than just shortening of the body. In addition, all chordates, at least at some point in their life cycle, have a muscular post-anal tail, and that also assists with this locomotion by lateral bending. So now we come to the very last branch before we get to the vertebrates, the Urochordates. So these are the closest relatives, the sister taxon, to the vertebrates. Are you ready to meet your closest relatives? I know you're really excited. Here we go. Oh no, not that guy. Really? Are you kidding me? This is a Urochordate, also known as a tunicate or an ascidian, and it basically looks like a blob of goo just kind of stuck to the bottom of the ocean floor. It doesn't look like anything that would be so special as to be a close relative to the vertebrates. How can you be telling me that this is a close relative of vertebrates? Well, to find out, let's take a closer look at the inner structure of one of these tunicates. So here's what they look like on the inside. Let me just walk you through how their digestive system works. So they've got an in-current siphon where they pull in water through this pharynx that's perforated all around with these pharyngeal slits. And we also see that they have an endostyle that's going to work exactly the same as the endostyle that you saw in the lancelets and the amphioxus. So it's going to secrete a mucus that's going to move down into the digestive tract with the little particles of food stuck in it through the stomach, up through the intestine, and into this area called the atrium and out through the excurrent siphon, along with all of the excess water that came out through these pharyngeal slits. They're all going to come out through the excurrent siphon. So we can see here, we've got pharyngeal slits, we've got an endostyle. That's a good start, but really, amphioxus had a lot more chordate features than that. That doesn't even really have all of the chordate synapomorphy. So how can this be the closest relative of the vertebrates? So the key is that all chordates have all of these synapomorphies at some stage in their life cycle. So let's take a look at the Urochordate life cycle. They start out, as we all do, as a fertilized egg, and that egg develops into something called a tadpole larva. And so this tadpole larva is free swimming and feeding, and then eventually it finds a good spot to metamorphose into this adult sessile form that attaches to the ground. They're actually often colonial, so they'll have attachments to other members of their colony. And if we look a little bit more closely at the morphology of this tadpole larva, we see that they do in fact have a dorsal hollow nerve cord, they have a notochord, and they have a muscular post-anal tail. All of this equipment here is exactly like what you saw in the adult. So it's got an in-current siphon that goes through these pharyngeal slits, into a stomach, an intestine, and the water and the waste come out through this excurrent siphon. So just another view so you can see this a little bit more closely. Here is the endostyle looking through that head portion of the tadpole larva, these pharyngeal slits in the pharynx going out to the tail, where we see a nerve tube and a very big notochord that stiffens that body, along with these lateral caudal muscles similar to what we saw in the lancelet in Amphioxus. So now we have a pretty convincing case that these Urochordates are in fact chordates. They fulfill all of the requirements of being chordates. But why do we think that they are the closest relatives to vertebrates? I mean, the cephalochordates really seemed a lot more vertebrate-like. And in fact, way back when I was first taking vertebrate zoology in college, we learned that cephalochordates were in fact the sister taxon to the vertebrates. But we need to always remind ourselves that science does not aim at establishing immutable truths. What you are learning in this class may turn out to be upended by future data. So its aim is to approach the truth by successive approximation. So every new piece of information gets us a little bit closer and a little bit closer to the actual relationships between these organisms. And then without ever claiming that at any stage final and complete accuracy has been achieved. So we never stop asking questions, we never stop adding information, we always keep looking to refine our understanding. And so that's what's happened in this case. What was found has to do with a population of cells in vertebrates known as neural crest cells. So neural crest cells are migratory cells that appear early in vertebrate development alongside the nerve tube. That's why we call them neural crest. They appear at the crest of the nerve tube. And they migrate out throughout the body through these very, very distinctive pathways. So we've got a lateral pathway, a ventral pathway, and they're very, very precise, very repeatable. And these neural crest cells in vertebrates form all kinds of structures, mostly having to do with the formation of different structures in the head. So we get from neural crest ectoderm, peripheral nervous system, so all of the nerves outside the central nervous system, visceral skeleton, so that means that the gill arches and neural crest is very important in the formation of jaws. Cranial muscles, so again, very important in the formation of the head. Part of the teeth arises from neural crest, and also pigment cells. And what was discovered is a population of neural crest-like migratory cells in Urochordates. So what they found was that these migratory cells follow the same pathways that we see in neural crest cells that are found in vertebrates. They also form pigment cells, so they have a similar function, and it seems very, very likely that these migratory neural crest-like cells were in fact a precursor to the neural crest cells that are found only in vertebrates. So the synapomorphy that joins Urochordates together with the vertebrates as sister taxa are these migratory neural crest-like cells, which as far as we know are not ever present in cephalochordates. So we've got the four synapomorphies of the chordates, but I usually like to think in terms of the big five features of all chordates. And I include pharyngeal slits in this list because although they're not a synapomorphy of the chordates, they're also present in hemichordates. They are critically important to the lifestyle of the chordates, and they're going to tell us quite a lot about the lifestyle of the earliest chordates and then subsequently the lifestyle of the earliest vertebrates. And then, in addition to that, the four synapomorphies: endostyle, notochord, dorsal hollow nerve cord, and muscular post-anal tail. Be sure that you really have a clear understanding and have committed to memory the functions of all of these features so that we can begin to talk in class about what these features can tell us about how the earliest chordates might have lived.